Life. The uniqueness of our planet Earth. There are animals, birds, a variety of fishes, insects, plants which make our cities greener, and forests that act as major oxygen generators. But the oldest creatures on Earth are the microbes. Scientists believe that Earth may have developed conditions to sustain life around 4.3 billion years ago. However, the oldest human fossils are only 3.7 billion years old. The evidence of microbes was preserved in the hard structures called stromatolites, which date back to 3.5 billion years ago. Stromatolites are created as sticky mats of microbe traps and bind sediments into layers. When cyanobacteria evolved about 2.4 billion years ago, they set the stage for a remarkable transformation. They became the Earth's first photosynthesizers, making food using water and the sun's energy, and as a result, released oxygen. This catalyzed a sudden and dramatic rise in the oxygen levels, making the Earth's environment less hospitable for other microbes that could not tolerate oxygen. Evidence for this great oxidation event has been recorded in the changes of the seafloor rocks. Microbes live in every kind of habitat, be it on land, in water, in atmosphere, or any living host. Microbes are truly the rulers of the Earth, evolving since planet's nascent stages till date. They have acquired almost all spaces on the Earth, like soil, air, caves, deep sea, salt lakes, deserts, hot water springs, animals, plants, and even on humans.
Microbes are those tiny organisms which we cannot see with our own eyes, but they are present everywhere. And when I say everywhere, it means our body also. Almost every part of our body is occupied by microbes.
An adult human body hosts complex microbial communities of bacteria, archaea, eukarya, which are commonly referred as human microbiota. And the genetic repository of these microbes is together called as human microbiome. Studies using advanced techniques like DNA sequencing platforms have significantly expanded our knowledge of human-associated microbes. It is estimated that a human body is inhabited with more microbial cells than the actual human cells. Microbes in our body may impact our physiology and development. Factors such as the host genetics, diet, geography, and ethnicity contribute in shaping human microbiome.
So today the microbiome is associated with almost every aspect of our body, including our intelligence, our memory, our moods like depression, anxiety. Almost every aspect of our behavior is affected by microbes.
The bacteria in the intestine are more than 100 trillion. Some are good, some are bad, and some are indifferent. These bacteria basically grow on food that we eat, which we do not absorb. Therefore, the diet, as well as our genetic makeup, as well as the environment, determines what is the composition of the bacteria that we have in our intestines.
The importance of human microbiome has prompted many countries to initiate mega projects to understand human microbiome in their communities. Consider the European Commission funded Metagenomics of the Human Intestinal Tract, or MetaHIT, or the National Institute of Health funded Human Microbiome Project, HMP. These studies have contributed immensely towards our understanding of the structure and functions of human microbiota. These projects have provided proofs of the roles of human-associated microbes in human health and a range of diseases. Studies on the Western populations suggest a strong association of microbiome with the general health and also with diseases related to neurology and gastrointestine, all the way to lifestyle-associated disorders like obesity, diabetes, and even cancer. But these studies have also shown that human microbiome is not similar everywhere. Microbes in and on human body are different in different geographical locations and communities. Latest research has shown that individuals from specific geographical location having similar dietary and cultural practices show similar gut microbiome. India is a geographically and culturally diverse country. Scientists are curious to understand the human microbiome of diverse Indian regions. The Department of Biotechnology, Government of India, has started the Indian Human Microbiome Initiative, the IHMI, the first ever mega survey to understand unique microbiome of Indian communities. The multi-institutional project is a collaborative effort of National Center for Cell Sciences, Department of Anthropology, the School of Health Sciences of Savitribai Phule, Pune University, KEM Hospital Research Center, All India Institute of Medical Sciences, New Delhi, the Institute of Bioresources and Sustainable Development, Imphal, the University of Transdisciplinary Health Sciences, Bengaluru, and the SRM University in Tamil Nadu.
Indian Human Microbiome Initiative is a very unique initiative where not only the microbiome of some 17 communities residing in 4 different biogeographic zones of India is being studied, but in addition, lot of information about their lifestyle in terms of diet and other activities is being collected. And also one important component of this project is to derive genetic information of at least some of the individuals participating in this study.
India, as the world's most populous country, boasts of remarkable cultural and traditional diversities. With over 705 indigenous ethnic groups, and each preserving its unique heritage, the country is a mosaic of varied traditions and customs. To represent the extensive population diversity, 17 different endogamous communities were selected for studying the microbiome of India. These 17 communities are spread across Ladakh, Haryana, Uttar Pradesh, Assam, Meghalaya, Jharkhand, Telangana, Maharashtra, Karnataka, and Tamil Nadu. To capture the overall microbial diversity from these populations, the oral, stool, and urine samples were collected in 2 different seasons. These samples were further analyzed for its DNA content, and the genetic diversity of the microbes were determined using high-throughput DNA sequencing technology. Now, this huge data is being further processed using bioinformatics to generate India's first comprehensive healthy microbiome map.
Criteria for identifying communities is based on again the cultural practices and the biogeographic zones. So India is rich in biogeographic zones. We have 14 different biogeographic zones, and geography, age, diet, lifestyle, everything actually contributes to the development of microbiome. From every zone, we identified at least 3 communities, minimum 3, one which is vegetarian, one which is non-vegetarian, and one which is tribal. So that way we carefully selected, with the help of anthropologists, 17 different endogamous groups.
Endogamous groups are the population where marriages happen within that particular group. It's a culturally defined boundary, but it has biological effects. Marrying within the group defines genetic variation within the group, which has certain genetic implications. In the Indian endogamous groups, dietary and culinary practices are special and unique. Spices, cooking oil, recipes, cooking time are all similar within specific endogamous groups.
Anthropology is basically a holistic study of humankind. So when I say holistic, it means organic evolution of human beings, their current biological variation, as well as the cultural and social practices of all human societies in the world. So in this context, it becomes very important in microbiome study because our diet is affected not only by what is available in our ecological setting, but also because of our cultural practices and belief systems. For example, you may get rice in certain region, you may get meat along with all other your cereals and other things, but whether it is part of your diet or not is decided by your cultural practices. Some people are allowed to eat meat, other people are not allowed to eat meat by their cultural beliefs.
Diversity in Indian geographical regions and the communities is also reflected in dietary practices. In South India, the most common cereal consumed among people is rice, which is used as an ingredient for various dishes. Most of the southern states have a large shoreline and hence they are inclined towards seafood. Wheat, along with the rice and millets, forms the staple diet of North India. Wheat is usually served in the form of rotis and chapatis, along with saag, bhaji, and tarkari. The northeastern states have unique vegetarian and non-vegetarian diet, while the central Indian meal is combination of northern and southern Indian recipes. IHMI scientists are curious to know whether traditional Indian diet represents specific microbiome of the particular region. Another unique element in the study is Ayurveda. The ancient medical system is based on ancient writings that rely on a natural and holistic approach to physical and mental health. Ayurvedic medicine is one of the world's oldest medical systems and remains one of India's traditional healthcare systems. IHMI will also try to find the association between microbial communities in the gut and 3 distinct Prakriti types.
The component of Ayurveda, what we are adding to this project is very unique. Every physiological mechanism has a diversity or variation in population, and Ayurvedic variables or Ayurvedic concepts, they help us understand this variation. For example, we are including Prakriti aspect in this project. Prakriti is human constitution or phenotype as determined by Ayurveda. We all can be classified into Vata, Pitta or Kapha Prakriti, and each Prakriti has its own response to external stimuli or the environment.
Vata, Pitta and Kapha are 3 doshas in Ayurveda that determine a person's health. These doshas are dynamic energies that are constantly responding to a person's thoughts, emotions, environment and actions.
We are studying whether the microbial signatures, they are specific to Vata, Pitta, Kapha Prakriti.
In the last 3 years, sample collection and the processing of the samples in laboratories have been completed. Now, analysis of the huge data is under progress. But this was not an easy task. Volunteers across the country had to be trained exclusively for the sample collection.
The most important thing is the training part of it. And here we had not only the NCCS team, but also teams across India to train. For us, we did a centralized training for entire teams. All the teams from India gathered in NCCs, you're in Pune University, and we conducted training. We conducted two sorts of training. One training was purely technical training, which was given by the NCCS and the Department of Anthropology, University of Pune, who gave training for the anthropometry. The other training was actually training people to just go into the community. People feel it is easy because I live in the community, I go, but it is not that easy. Asking people to imagine, give your stool, that itself, people start this way. How do you convince people? How do you tell people? We have to tell them, but to tell the people, every researcher has to be trained meticulously.
The another challenge which we faced on the community is we have selected families with the individual of age group 18 to 55 years, and we have selected only one member of the family for the study. But we have witnessed few families where there were multiple members of that age group. So it was very difficult to explain them why only we are choosing that particular individual and not others. So convincing the family that we are going to recruit only one participant per family was little bit challenging. But again, the experience and the rapport with the community has made these things possible for us.
Overcoming all these challenges, large number of samples have been collected and enormous data is being generated as a result. Scientists are using advanced technology and unique methods to analyze this big data.
We have collected more than 4,200 samples from 3,400 individuals. So we collected 3,400 samples from the first time point, and the remaining samples were collected for the second time point, just to check whether the seasonal variation has any impact on the microbiome. So for all these samples, we did amplicon library sequencing, amplicon sequencing. It means that we are sequencing a particular gene from a particular microorganism. So this gene helps us identify that particular microorganism. From every individual, we did 4 amplicon libraries. So microorganisms, they belong to different kingdoms, right? So one is bacterial kingdom, second is fungi, third is microeukaryote, and fourth is archaea. So for all 4 kingdoms, there are certain genes which are used for identification. So from bacteria, we sequence approximately 1 lakh reads per sample. Same way for archaea, for microeukaryotes, and for fungi. So you can imagine, from one individual, from one sample, we are sequencing approximately 400,000, that means 4 lakh reads, and each read is representing one particular microorganism. And then you can multiply that with 4,000 individuals, so you can imagine the scale. And this is just amplicon library sequencing. For again a subset of individuals, we also did whole metagenome sequencing. So amplicon library sequencing helps us in identification of microorganism, but when we do whole metagenome, it also helps us identify what are the functions these organisms are doing in that particular individual.
The Indian Human Microbiome Initiative was envisioned with an ambition to study the gut microbiome of Indian communities. At the ongoing analysis stage, scientists are confident about the outcome of the project. This may lead to novel scientific discoveries as well as applications useful for the human health.
Stool from a healthy person with a healthy, more beneficial bacteria can be transplanted in a diseased individual. That's called fecal microbial transplantation therapy, or FMT. And it has been seen in certain disorders. So over time, there's a lot of excitement about this, and this kind of treatment may become a reality for many diseases.
The most important outcome of this study is going to be our understanding of what we call as baseline microbiome or healthy microbiome of the communities that have participated in this study. So microbiome holds tremendous potential for offering gentler, milder, individual specific and cheaper solutions for a variety of diseases, including metabolic disorders like obesity and diabetes, neurological disorders like Parkinson and Alzheimer, and in general for well-being of all of us. Another important aspect of this study is the study also includes some tribal communities which do not have exposure to any modern lifestyle. So, that will also give us information about what we can call as virgin microbiome, where this microbiome we expect that it would not be affected by any of the modern lifestyle like food or exposure to antibiotics, and this information would be very useful in understanding how the modern lifestyle has affected our microbiome. At the same time, the study also involves generating some bacterial cultures from the individuals that have participated in this study, and these cultures would then be very useful to develop probiotics, because the modern lifestyle is destroying the healthy microbiome that we have, and maybe after a few years we will be losing entirely the good healthy microbiome that we have. So, these cultures available in the repositories would come for the rescue, and we can recolonize our unhealthy microbiome with these healthy microbes that are present in the culture collections.
What are the unique features of microbiota of Indian population? Is it possible to define core Indian microbiome? What is the association between age and microbial composition of Indian population? How does the changing lifestyle and diet in the current generation affect microbial composition? Is there a possibility to develop indigenous microbial consortia to tackle lifestyle associated diseases like obesity, diabetes, and cancer? Soon, we shall get answers to all these questions. The Indian Human Microbiome Initiative will be a path-breaking study not only for the Indian scientists but also for the scientists all around the world.